Cell Membrane and Transport by Alexa, Kyra, Mahi, and Anna. Phospholipids make up the basic structure of a cell membrane and consist of alcohol and fatty acids. There are two main components of a phospholipid, its head and its tail. The diagram in front of you is a fluid mosaic model of the membrane and is held up by the phospholipid bilayer. The head of a phospholipid is hydrophilic because it loves water and points toward cytoplasm or whatever fluid is in the cell. The tails of the phospholipid are hydrophobic, meaning it hates, or more technically means it repels water. And because it makes up more of the majority of the phospholipid bilayer, small hydrophobic molecules are allowed through, unlike the polar hydrophilic molecules which need extra help. Aligned within and outside the membrane are various structures. Glycoproteins are useful for cell recognition as they act as receptors. Glycolipids, which are attached to a carbohydrate, assist in cell-to-cell recognition. Cholesterol maintains the fluidity of the cell surface membrane on the outside. On and inside a plasma membrane, there are various discernible proteins. They are prominently classified in two categories, peripheral and integral. Peripheral proteins, like certain hormones and G proteins, are found on the surface of a membrane and can detach from it without changing the membrane. Their main functions include acting as receptors, enzymes, and facilitate movement through other components. Integral proteins are found within the bilayer. They function as transporters in the membrane and act as pathways for ions and molecules. Broad categories of integral proteins are carriers and channels.
So the cell membrane has many functions, but some of the most important ones are that it protects the interior of the cell by only allowing certain substances into it while keeping others out, and this is known as selective permeability. It also helps to support the cell and maintain its shape. It regulates the cell growth through the balance of endocytosis and exocytosis. It serves as the encasement of internal organelles, and it can also serve as a base of attachment for the cytoskeleton in some organisms and the cell wall in others. So this picture shows some more specific activities of the cell membrane that contribute to its overall functions of protecting and maintaining the cell. So this includes the transport processes, signal detection, cell-to-cell interactions, and all these activities help the cell membrane carry out its overall main function of protecting the cell. All cells do have an outer plasma membrane, but unlike prokaryotes, eukaryotes possess internal membranes.
In passive transport, substances move across a membrane without using energy directly. All types of passive transport involve diffusion. Diffusion is the movement of a substance to an area where it is less concentrated. During passive transport, molecules move down the concentration gradient. If they diffuse long enough, the concentration gradient will eventually disappear. A concentration gradient is different concentrations of a substance on either side of the membrane. Some examples are simple diffusion, facilitated diffusion, and osmosis. In simple diffusion, substances can pass through the membrane freely. They move down the concentration gradient without any help from carrier molecules. Nonpolar molecules like oxygen and carbon dioxide can easily diffuse across the membrane. In facilitated diffusion, substances can diffuse through the membrane with the help of protein channels. The channels move certain and specific molecules across the cellular membrane. In active transport, energy must be used. This is often in the form of ATP. It moves solutes from a high concentration of water to a low concentration of water. So, from low concentration of solute to high concentration of solute. The cell uses a transport protein to move the substance against its concentration gradient. Some examples of active transport are protein pumps, endocytosis, and also exocytosis. Endocytosis allows cells to engulf fluids and large molecules to bring them inside the cell. Part of the cell membrane pushes inwards and traps the particles. A vesicle forms around and brings particles into the cell. The vesicle surrounds the particles inside of the cell. Pinocytosis and phagocytosis are the two main forms of endocytosis. Phagocytosis is known as cellular eating, and penocytosis is known as cellular drinking. Exocytosis is the opposite of endocytosis. A vesicle surrounds the cells inside the cell that need to be exported. The vesicle moves to the cell membrane and then fuses with the membrane. It then releases the particles outside of the cell.
The term hypo, meaning low, and hyper, meaning high, refer to and describe the solute concentration outside of a cell. In a hypertonic solution, the solute concentration is higher outside of the cell than inside, which also means that there's less room for water outside, resulting in a lower water concentration outside and a higher water concentration inside. Conversely, in a hypotonic solution, the solute concentration outside of a cell is less than inside, which means there's more water concentration outside and less water concentration inside. Because of water's nature to go from a region of high concentration to low concentration, if a solution is hypertonic, meaning there's less water outside of the cell, water will naturally leave the cell. And if a solution is hypotonic, meaning there's more water outside of the cell, then water will enter the cell. Each of the 6 bags in our experiment, we were told, either had a mole of 1, 0.8, 0.6, 0.4, or 0.2 sugar solution. And once we placed them in their own beaker of water, each beaker of water had equal volumes of water. Our purpose in this experiment was to find the difference in weight of all of the solutions before and after they had sat in the water. The bag that is the most hypotonic would be the one that gained the most water, because hypotonic solutions suck in water. After we examined the solutions after they had sat in the water, we could use math to figure out which had gained the most water, but also we could observe that some solutions felt firmer in their bags and others that felt more limp. So we used our sight and our touch and math to figure out which bag would be the most hypotonic and which bag gained the most water. We also found that one solution in the bag had, in fact, lost water, and we determined that this solution was hypertonic, because in hypertonic solutions, water leaves the cell.